Hi everyone, it's Mr. Sinti, and today I have the pleasure to talk to you about how plants can affect their movement, they can affect their growth rates, and other physiological responses due to cues from the environment. And in particular, the most important cue that plants can get from the environment is light. But there's some other factors at play too, as well. There's something called circadian rhythms that control this as well, and there's other environmental factors as well, like for example temperature and amounts of carbon dioxide. But most importantly, it's light. Light seems to be providing, as you can see, this daffodil. Light provides a lot of cues that plants use to adjust their growth and whether or not they germinate or whether or not they flower. And so this is the way plants respond in their life to changing environment. Bear in mind that plants are indeed planted and they cannot move around. They cannot show so much like giddy up in terms of migration or other sorts of responses. So this is how they maintain it. And so let's jump right into that conversation and see what comes of it. And so we call this big word photomorphogenesis. You like that? Photomorphogenesis. So using light as an important factor to make structural changes, morphological changes, so physiological responses. So in addition to light being critical for photosynthesis, it's also important for plant growth and development as well. And so you can see these beautiful sunflowers here. So light can affect their morphology. In other words, they can grow toward the light, phototropism. And so plants are able to detect the passage of days, the passage of seasons. They can detect certain frequencies of light and also intensity. Pretty intense. And so, as it turns out, when you look at just the simple green pigment, chlorophyll, that we find often in plants, in their leaves, in particular in the palisade, or spongy mesophyll in the parenchyma cells, chlorophyll, green pigment, it's green, remember, because it doesn't absorb green. What it really absorbs is red and blue, red over on this side and blue over on this side. And so they like red and blue in particular. And green is not absorbed, so therefore it reflects back out. So plants can detect direction of intensity wavelight. We call this the action spectrum here, where you can see white light can be separated into different frequencies of light in nanometers. So each one's sort of monochromatic. And you can shine this and you can measure absorption using a spectrophotometer. And you can see here that chlorophyll really likes blue and it really likes red. And so it turns out there's a lot of processes that plants do in addition to photosynthesis, their growth and their germination. And it seems like there's more than chlorophyll as a pigment. There's many other pigments that plants have, especially in their leaves. You might suspect that, but they have pigments in their leaves that can detect different frequencies and then they can make adjustments. And so as it turns out, the two colors of the rainbow that plants prefer are red and blue. And so these observations have led botanists to classify a lot of these groups of pigments that are capable of photomorphogenesis in plants as blue light receptors. In other words, pigments that really like the blue. This is chlorophyll, but I'm just saying blue light receptors really pick up the blue, and then there's these red light receptors called phytochromes that really prefer the red light. And then depending on which pigment is picking up which, the plants can make adjustments, and we're going to look at that in this video. So what's fascinating right out of the gate is phototropism, as you can see the bending toward the light down here. Do you notice if I were to ask you, which color, if you were able to shine just red or orange or just yellow or green, I can ask you, does yellow light cause phototropism? Now, we all know that light causes phototropism, but light is broken up into the rainbow of colors. If you're able to isolate it and just show one color, as you can see, red doesn't do anything, or orange or yellow or green, but blue light does. So it turns out that blue light photoreceptors, a bit of a detail, but it's an interesting point. The blue light photoreceptors are what are picking up the blue light and therefore creating phototropism. You may recall that even though that pigment is being detected by blue light photoreceptors, ultimately it's the hormone auxin that's going to be causing phototropism. So it's a combination of picking up a signal and then a hormone transmitting that to the cells, which increase growth and also water accumulation on the opposite side of the stem, which causes it to bend over. So it's kind of coordinated. So plants really show incredible physiological responses like phototropism based on light. So this is what we mean by photomorphogenesis. So phototropism is an example of that. So when scientists tried to locate these blue light receptors, try to isolate them, crush up the tissue, sort of chromatography, separate chemicals, they were kind of elusive. They were very hard to find. And that's one thing. Eventually we will find them, but the names sort of appear in the literature as these blue light receptors are kind of cryptic or hidden, so they became known as cryptochromes. I find that to be kind of interesting. Again, a detail. This plant right here is probably the most highly studied plant in the whole plant kingdom, which is Arabidopsis. It's a type of mustard, and we know so much about this. So many studies have been done on this. Mutants, wild type. It's sort of the Drosophila melanogaster of the plant community. We've sequenced every gene. We know a lot about this, and so we were studying mutants of Arabidopsis in order to find what's going on with these blue light detectors, receptors. And so as it turns out, these cryptochromes, or blue light receptors, can affect hypocaudal elongation. The hypocaudal, hypo, remember, means below, the cotyledons, and epi is upon. It can affect that. That's one of the physiological effects of the blue light receptors. As I mentioned before, the blue light receptors can also affect phototropism. They can also help to regulate the opening and closing of the stomata. And so the red light ones are called phytochromes. And phytochromes, the fact that they are red light receptors, like red light. So when you shine red light on them, they're capable of doing things. And so early studies in lettuce seedlings. I don't know if you've ever considered that lettuce, I know you buy this at the store and you make salad out of it, but it starts off as a little tiny seed. Lettuce seedlings are very tiny. And as it turns out, it's kind of common for plants that have small seeds like lettuce. You don't want to germinate. In other words, begin your growth if the conditions are not right, if there's not enough light. And so they're sensitive to this. And so when the certain amount of light comes, they're able to start germinating. And so I just want to get into this. It's kind of interesting. And I just want to drive that point home that, like, for example, if you're in shady environment, maybe that's not a good time to start growing. Does that make sense? It's like you won't get any light and you won't make it. But maybe, how about this? Maybe there's seeds on the ground, and then when a tree falls and then there's more direct sunlight, then it's time to germinate. And so how does it all work? And so an investigation began in the 1930s, so a while ago, by the Department of Agriculture, and they were trying to see like light-induced germination. So in other words, like here are all the different colors, and here are the wavelengths. And so you could shine different frequencies of light at these lettuce seeds. And so as it turns out, let's shoot violet at it, nothing. Let's do blue, nothing. Let's do green, nothing. Yellow, nothing. Orange, nothing. And as you can see here, when you shine red light, they're able to start to germinate. And of course, they do not germinate in the dark. And so, so what? Okay, so they like red light to germinate. But then check this out. When you crank up the frequency just a little bit more, so in other words, pure red is 620 nanometers, and then just on the fringe of not being able to see it, like infrared, it's called far red. I know you're going to ask me what's the difference, like visually, between red and far red. I can't tell you in terms of that, but I can tell you that it's a longer frequency. I hope that satisfies you. So if you shine far red, check this out. What's happening? So red means germinate, and then you add far red to this, and what happens? It stops germinating. Interesting. I wonder what it means. And then over here, red means to germinate, then far red means don't germinate, and then red. So like we shine red, far red, and then what do we get here? Germination. What do you make of that? It could mean that perhaps it's the last light that hits it that sort of invokes this. But it's certainly one thing. It tells you that it can go back or forth. And then look at this sort of torture here. Red, far red, red, far red. So the very last one is far red, so therefore no germination. You're like, well, this is all well and good. Like when red light increases germination and far red inhibits it. And you're like, okay. And so it's sort of dependent on the last flash of light that gets it. And so that observation is interesting, but the more profound question is, why does the plant respond to this? I mean, what advantage does it have differentiating between like far red and red? Why is it good sometimes to germinate, not good? So obviously the plant has an ability to control this, and so this is the overall question in our head right now. What's the importance of this? So as it turns out, further study indicates that these phytochromes, these red light receptors that like red light, are a combination of a big protein, actually two proteins right here. So they're identical proteins, and as it turns out, this little patch right here is called the chromophore. The chromophore is the part of this phytochrome that actually picks up red light. So it's the pigment inside the protein that picks it up. And I just wanted to establish that. It's a kinase enzyme activity that results in this phytochrome. And so check this out. As it turns out that depending on whether light, let me see if I can illustrate this. When red light comes down, like this, here's red light. When it comes down like this, it's detected by this pigment. And if you recall your biochemistry, sometimes when a frequency of light strikes a molecule, it's capable of changing it temporarily, so it'll alter its shape. And if the pigment alters, if the chromophore changes its shape, then the whole protein will change shape. And so then when a protein changes shape, you can get something done. So come back to that. So here's some terracotta pots. Over here, light is coming down here. It looks like, oops, we planted the seeds a little too deep, and so they're not being able to pick up the light. And so, therefore, not enough light is penetrating, therefore no germination. Oh look, here we planted them just a little bit below the top of the soil, so light is penetrating, they are able to germinate. So that's showing that. And you're like, well, how does the seed know? Well, it picks up red. And you're like, really? Yes. It picks it up by its photochrome. And the photochrome, remember, is this combination here of the protein and the chromophore. So the protein is shown here. You can see here the beta pleated sheet and the alpha helix. So it's a protein with this chromophore light-sensitive pigment, which is shown here, these four rings. And so when red light it comes down like that and strikes it, it changes its shape, therefore it changes the whole phytochrome shape. You're like, well, what's that going to do? Well, this is a very important picture. I don't know what your take on it is, but it sort of reminds me of when I say change shape, it kind of reminds me of, no pun intended on this, but it's because we're talking about light, but it reminds me of a light switch being off or on, up and down. It's changing positions. And so check this out. When it sort of can convert from one to the other. So when red light comes down, like this, here's the red light, it's coming in. The phytochrome picks it up and it alters its shape, and therefore the protein changes its shape down here, changes its, see how the protein is sort of closing up right here, like rabbit ears. It's getting a little tighter. So it's changing shape, and that results in some responses. There's like a cascade within the cells that carries out seed germination, flowering, and so these are the physiological responses. So red light initiates that. But then when you shine far red light on it, far red light converts it back into this other conformation and therefore shuts it off. So on and off, on and off. So the exposure to come away from this, the exposure to far red light inhibits. The exposure to red light encourages. So you got a plus and a minus. I haven't really mentioned why this is significant to the plant other than it's capable of doing it. And so do you see here there's something called PR and PFR. So what this is in reference to is the phytochrome R and the phytochrome far red. So phytochrome R can receive red light, and therefore it turns into this conformation, which is called the PFR, which in this conformation, it picks up far red light, and then it converts back over there. That's what this is all about. And so, get rid of that. So, red light on, far red light off. And so, as it turns out, here it shows it again. The PR prefers the red light, which is at just slightly shorter nanometer. This is at the brink right here. So this causes response. This causes to be inhibited. So make sure that you understand the differences between those two. And so you can, again, this is another example of getting back to those lettuce seeds. Like when you shine red, they germinate. The dark, they don't. Far red, they don't. Far red, red means they do. Far red, or red, far red, they don't. So it's the last flash. And so basically, what is this all about? Why do plants need this sort of conversion? Well, it tells plants about the quality of light. That's very important because, the truth is, during the day, if you're in direct sunlight or whatever, there's sort of a mixture of far red, red. And so it's sort of a teeter-totter between the PR and the PFR. So it kind of goes back and forth, dynamic equilibrium, sort of just rolls. But being able to roll, in other words, plants can use these ratios. If there's a lot of red or a lot of far red, you could use that to sort of adapt to whatever the conditions may be. You can roll with this because, once again, the PR prefers the red, that turns things on. The PFR prefers the far red, that turns things off. So here's the punchline of this whole thing. So if a plant is growing in the shade of others, like in a forest, like what happens is that a tree might want to change its growth pattern based on light intensity or quality. And so as it turns out, if other trees shade the tree, its phytochrome ratio shifts in favor of PR. In other words, the canopy screens out more of the red light. So you want more PR to be able to capture more of the red light than the far red light. And so therefore, if it can capture more red, it's going to grow taller because it's in a shady environment. So it's a way in which the plant can put its sources into growing taller. But if you're like in direct light, for example in this tree, if you're in direct light, maybe there's a proportional shift like a teeter-totter to the PFR, which is the one that picks up far red better and therefore inhibits. And so if it inhibits height, maybe that's going to stimulate, for example, so it increases, which stimulates branching. And so you might get better branching this way, and you don't need vertical height. So you can capture more light that way. So it's a way in which, I know it's a little tricky, but it's a way in which subtly a plant can adjust based on whether or not it's getting a lot of light or not a lot of light. It can make adjustments using its red, far red. And then I mentioned this at the beginning of the video that some plants are controlled, interestingly enough, in terms of their responses, whether or not they they open and close their stomata, whether or not they conduct any physiological response, not by any outside cue. It's almost as if they internally know what's going on. It's called an internal biological clock that controls what scientists called a circadian rhythm, which is circa about a day, around 24 hours. And so this is weird. Transpiration can oscillate. In other words, guard cells can open and close even if you keep them in the plants in a growth chamber and you don't show any light to them and you keep the conditions perfectly the same. So there's no factor. When you keep them in constant condition and they're still able to do things, the only explanation is that it's some kind of biological clock that's controlling it. And so when you keep plants in a dark chamber, they still continue to like open up their leaves or close their leaves as if they, like if it's in the morning or if it's in the evening. It's kind of fascinating. And so we call that circadian rhythms. And let me just give you an example of what I mean about opening and shutting these leaves, because that's kind of abstract. And so this is what I mean. Here we're shining light on them. And so obviously plants are able to make this sort of adjustment for light. I mentioned this before, like phototropism is the blue light receptor. But if you keep them in the dark, so picture this in the dark. I cannot show you this in the dark because you wouldn't see it. But picture the plant doing this in total darkness, and you're like, "Whoa, what's going on? What the?" Well, it's some kind of internal clock that must be causing this to take place. And you're like, "What kind of?" That sounds a little bit magical. Internal clock. What do you mean internal clock? What is that? Well, the internal clock that I'm talking about is not magic. The internal clock that I'm talking about is something that must be controlled by chemicals. And so, getting to the bottom of this, what scientists did is, well, it's hard to control everything. And so we even put plants in satellites to make sure that gravity wasn't affecting them as much, or we put them in the shafts of the deepest mines to make sure that there was no factors affecting them. So it seemed like it was internal clocks that were causing this, and it seemed to be set to a 24-hour period. And so it's kind of mysterious. And so it turns out that processes that were once sort of regulated in a really tight 24-hour period, when you keep them in dark long enough, they seem to sort of move away from that 24-hour period. So in other words, they become synchronized, I meant to say. So they're not synchronized to the outside world, and so it fluctuates a little bit. I'm sorry about that. So you can take bean plants and sort of restrain them, like with strings and things, in the dark, and then you can let it go, and it'll resume right where it was supposed to at that time of day. So it really shows you that you can interfere with the biological rhythm, but the clockwork keeps on ticking. So there must be some sort of chemical inside. And so, as it turns out, that we don't know for sure, but the leading hypothesis is that there's probably some kind of protein regulating through a feedback control mechanism. And so let me get into that just for a second. There's probably a protein that's a transcription factor. And so transcription factors can either promote or inhibit transcription. So this particular protein inhibits the transcription of the gene that encodes for the transcription factor itself. So as the transcription factor accumulates during the first half of the day, and then it'll ultimately decline in the second half of the day because this itself inhibits its own production. So it's kind of like an hourglass. It's like the more you make of this, then it becomes this, and then it becomes this. And so it oscillates back and forth like that internally because of transcription factors. And so what's fascinating finally is that we've been able to take a gene, and here's this Arabidopsis again, this mustard plant. We were able to take a gene that codes for this enzyme called luciferase. Now this is a very cool enzyme which causes bioluminescence in, like for example, fireflies. And so you're able to splice it into a promoter in a plant that shows circadian rhythms. And so as it turns out that on a cyclic manner, in a 24-hour period, it's like the plant starts glowing and then it doesn't glow, and then it glows and then it doesn't glow. There's a picture of this. And so it really shows, this is actually tobacco. We were able to splice it into tobacco as well. But this is kind of interesting that we're able to see biological clocks or circadian rhythms by the fact that they glow, don't glow, glow, don't glow. I mean, we could see guard cells opening and closing and leaves going up and down, but this is really interesting when you can make something glow, not glow. So I hope you enjoyed this video on photomorphogenesis and the whole detection of blue light receptors and red light receptors. Thanks for watching.